So good morning. Good morning for those who are away. Okay, so yeah, for this particular information about the research, so please go to the please again. Keep this information, and there's a couple of people you can email regarding your interests. So, for example, Sarah, she's having those meetings to discuss about research, different departments. Feel free to email and go to the links. Okay. So this is regarding research happening in the midterm. So the midterm, we have an issue in this room. And this room is not equipped with enough electrical sockets for everyone. And we don't want you to be in this room and suddenly get out of battery. So that's why we switched from the in-person format to the online format of the exam. And I think that will be less stress for you also to commute in the morning, and you can have like really more comfort to have it now online. But please read very carefully the announcement and the email I sent you, because if this is your first online exam, there are some technical problems will show up. Many times we tell the students that you need to have the respondents logged down. You have to download it in advance. And the respondents lockdown, it has also the ability to monitor your movements and stuff like that. So I'm not using lockdown. So is this your first online exam? No. Is this your first online exam with respondents? No. That's great. So you know how, you know the drill. I don't know to discuss that with you. That's great. For those who didn't, probably some of you didn't, please read the email and get your computer, laptop, desktop, whatever they use, tablet ready. And make sure you have and please make sure you have that set up in advance. Because during the exam, you don't want to stress that you are disconnected or kicked out from the system. Okay, and I'm remote. So I will be in my office, so I can't do anything. Then we will start calling IT and good luck. I think this might probably like 10 times you already reported that it's not working and still not working. And so the exam duration is going to be 80 minutes. And I was a student before, become a professor. So I know that day you will not feel that you want to get any lecture after. So that's okay. We're going to skip the lecture on that day. I would probably compensate by giving you a recording that week after, so you probably doing the reading week again also. Okay. For the session, I really recommend you tomorrow to be there, 12 to 1. I understand that office hours do not align with your schedule probably. That's fine. I will be recording that, but you will not have the chance to discuss back and forth. Yeah, and I'll post one preparatory quiz, just one question is not graded, just for you to go log in so that My Brightspace and your account in My Brightspace is active. You go there, just click it, see if it works so that on Wednesday, everything is okay. So in the first survey that is rated or survey quiz, it was two parts. One part was evaluation of how the course is going, and the other part was like just to go answer some questions and test your knowledge. So I take very good care of your responses, like, but sometimes I can't do it. Like, if I ask you, how do you think, what do you think about the slide content? And I get like many of you saying, oh, I would like to have less text. And then the other 50% would say the slides not worth it. If you, like, I can't really take the text out of them. I would minimize it a bit, but I still need to have it there. So the content, I'm gonna try to adjust the content based on what you said, but at the same time, sometimes it's so difficult to have a slide where you just make it as bullets, then add this, remove that, get to the result. You have to have some sort of sentence text, makes sense. And that's why you have sometimes you need more text. Alternatively, I will start to add more pages. Like you can see, I added in this module multiple pages. I didn't add too many, like really specific pages, so that you can go and read those pages to have a deeper knowledge of the. Okay, again, your exam is gonna be mostly the slides. For those who have much deeper knowledge,
Sorry, exam module and then to fill it.
yeah. So everything covered, the last slide I will explain today is going to be introduced in the part of the exam. Yeah. Probably I would remove some slides, but I will let you know. By Friday, you will know those information. Okay, the last thing is the assignment. So you guys have, I gave you the freedom to spread out yourself into groups. I added more diseases than the number of the people in the group. We are 102, so technically 10 diseases should be fine. But I added here 2 diseases so that you have the ability to choose. I didn't force you to make groups together. But I really advise you to work in group. If you have a chance, work in group. First of all, when you write a document and somebody else proofread it, it's way different from reading it yourself 5 times. So this is first advantage. The second advantage is that, like it or not, you will always work in a team. In your life, wherever you go, you will work in a team. So this exposure gives you a little experience of working with someone else. It might not be the best experience, but then you will know what are the positives and the negatives. In the later years, and we see that the groups become bigger, and we will work with 3 or 4 students. And I see the medical students most of the time they present a group of 5. So you can imagine you get along or not. And I'm giving you the lowest type of stress because I didn't prepare the groups myself. In other classes, the profs would simply take people with the first grade A, like 5 days, they go to. That's it, you can discuss it. So you have to work together. So anyways, long story short, if you are alone in the assignment, please try to match with someone else. And that's better for you. That's less workload. That's a better quality assignment. Okay, and at the end of the day, it's your decision. But please don't go in a group on your own. Leave the assignment in the last few days and give me a low quality assignment and then come back fight for the points. Okay, you don't put yourself in that situation. That doesn't necessarily mean that one person will make a low quality assignment and two people will do a better quality assignment. But that's our experience. We see that writing research, reviewing research with two people is much than one person. Okay? And now we will move on to today's lecture. Is going to be mostly discussing the DNA damage. And if I will have the time, we will discuss a little bit about the metabolic diseases. The last thing I want to announce probably is we will switch to online classes after the midterm all the way. It will be probably an easier way to document the information, record it, because I look at the recordings that I'm doing like here, it's really not. It's not really in a very the volume is not good. Some people are not able to come here. You in your surveys all the time ask to go more for the online. So probably we will switch to another person. I will announce that in an emails. So everybody. Okay. So far, we discussed almost all the biomolecules in the cell and how they are produced from the DNA level, RNA level, to proteins, in addition to biomolecules that we acquire, like copper, iron, and how those are included into functioning in the cell. So there is only one last part that we have to discuss is the metabolics. And you will see that these 4 lectures that we discussed, we will likely close in a loop right now at the end of this lecture, where we come back all the way to the first slides where we discuss a change of an electron from one molecule to another. It changes the function. So with this, you will have the deeper understanding how of that change of a one electron at the level of DNA, RNA, or protein could lead to a change in the function in the cell. And if there is an imbalance, that's definitely a discussion about going through an adaptation or a disease. So today's focus is mainly DNA damage, because last lecture we discussed how the cell prepares itself in order to divide. And we discussed extensively how it goes to 2 phases, where interphase it prepares itself, and then mitosis, meiosis, depending on the type of cell. But we didn't discuss what are the types of the DNA damage that happen there, and how can we correct those. So just a refresher. The cell would have checkpoints to make sure that the contents are adequately prepared for the cell to divide, as well as the cell size, the nutrient, the DNA molecule, the necessary proteins, and all that. So it will go through this interface where it will be checked just at the entry and just before the end of the interface, so that the mitotic process takes place. And if this is a gamete or like a sperm or an egg, then of course this is a meiosis process that was going to take place here. Now, the preparation of the genetic material is definitely the most important and the most critical step. And you could see from the end of last lecture that having a little bit more DNA or more genetic material during the division automatically translates to a disease, and having less of that gene or have that of that DNA is automatically translated to a disease. So it has to be accurate, and it has to be the exact same amount. And on top of this, we will need to have all the proofreading before and after. So you will see today that we will discuss how, if a mistake happened, that is a mismatch of a DNA, there is a system which can look into that and correct. Then we discussed how the genetic material is packaged into the nucleus, and of course, depends on the cell situation. The DNA material is going to be packaged differently. So at the time that we need to split the chromosomes into two, the compactness of that DNA should be the highest. Because you can tell separating many small things at the same time is more difficult. That's why you have these two systemical methods very well compacted, then it would be much easier to just split into two. Right? Now, this is where we started to discuss about an interaction between DNA and protein, where we spoke about these histone-like proteins, which are complexes where the DNA are wrapped around. And those histone proteins could be also post-translationally modified. Therefore, this is the protein and its superior level, the post-translation modification. And on the other side, the DNA itself could be acylated or methylated. So these post-translation modification on the protein are kind of translated into an epigenetic modification on the DNA. And of course, those play a role in deciding how the DNA is wrapped around those histones and if it's accessible for other proteins or not. And then it tells us that those interactions are very important in turning on and turning off the gene. So for instance, we discussed here how acetylation is associated with the process of activation of the gene transcription, while methylation turns off that gene. And the accessibility for that transcription factor to bind to that DNA doesn't happen anymore. So it's only one simple modification that happens at this DNA that causes the shutdown of the gene. And epigenome and epigenetics is one of the really hot spots in the last 5 to 7 years, where there's tons of these methylated acylated DNA are discovered to play a role in the regulation of the gene. So even if you have the whole machinery where you have all the transcription factors available, produced, and ready, simply having an acetyl or methyl group would block or turn on the gene. Okay, and today I will show you one other level of complexity, which is probably it's very, just to phrase it correctly, it's very well known in bacteria that the DNA replication is taking place. But probably if there's any issues that show up and mismatches, then the DNA polymerase has to correct them, right? But in order to know which one is the parental DNA that is the correct part, and the newly generated strand, it could be read by looking at the history of the methyl groups. So the parental groups would have the methyl, while the newly generated ones, they do not have that. And this is how probably the DNA polymerase will be able to say, oh, this is the parental strand, so this is my template, it's methylated. This is the newly generated one, it's not methylated, so I have to correct that one. Okay, so this is another level of complexity that comes from the epigenetic modifications. I will be describing in some of the slides. Then we move to the replication process. I didn't really describe all the history of the DNA replication. There's many theories. How did they come into that DNA is semi-conservative replicator. So I'm just giving you mostly the molecular aspects of stuff. Now, the DNA replication, we discussed that it's a very complicated step. It has to start at certain point. We need a lot of enzymes to be accumulated in order to get that DNA replicated. Some of them related to the topology of the DNA, because the DNA is like two winded strands. So you first need to unwind it. So that's a lot of energy. And then you have two strands. One has to go continuously in one way, and the other one, it has to all the time do simple lag phase. So these are small fragments. So on the upper strand, you will see the leading. So the replication is going that side. And therefore, this is continuous for this upper strand. As we unwind this DNA, it will continue reading all the way till the end. But on this side, if I'm reading, I'm going this way, well, I will reach the end of that fragment. And then the DNA is going on that side, that fork. So I have to go back all the way. And then again, start here. And I generate another lagging strand fragment. So there's one fragment I would imagine from that board to this one, and then another fragment here, and then another fragment there, and then all the way. So we will have a lot of these fragments generated on the lagging strand. And now this forces the replication fork to have more proteins to be recruited, which has to link those fragments together. So the ligases will take that fragment and ligate it to this one here, and then it goes all the way. It has to ligate this one with that one. And then it keeps going with the fork and keeps ligating those fragments. And these are the Okazaki fragments. Okay. Well, you can tell that's a lot of energy. That's a lot of proteins that are complexed together in that replication fork. If you open these, unwind these DNA fragment like strands, they tend to wind back together. And that's why you would have these proteins sitting here in order to avoid this. So in this one here, probably we have, the list of the proteins that we are talking about. And you can see the single-stranded binding protein, and these are the proteins that will keep the fork open so that the two DNA strands are unwound. Okay. Then we discussed, okay, great, we did the replication, we have the DNA material ready. The size of the cell is great, we can divide it into two. Then the cell is going to decide where to go. Based on if it's somatic cell, it's going to go through mitosis. If it's a gamete, it's going to go through two different steps. One that ends up in a similar way to the mitosis. So the last part of the meiosis is very similar to mitosis. But the first part is where the genetic material will make a mirror image. The chromosome 1 will come and like stick there, and they will exchange material. And then after that, they will be split. And if you look at these images here, by the end of the first round of the meiosis, you will have the red and the yellow chromosomes exchange parts of DNA before being split into the daughter cells. And the last difference between the mitosis and meiosis is that this cell has generated two identical cells with the exact amount of DNA, while the meiosis has generated 4 total cells with n equal 1, and the process is going to be complemented by the fertilization process. So one egg from male versus female merge together, come back to the n equal 2, and generate an embryo. Okay. So this is the story. We're kind of closing the molecular aspects from the DNA to the proteins, like all the way from DNA to RNA generation, to splicing of RNA, and then generating different proteins. But all the time, we have to keep in mind that the first theory of one gene, one protein is always valid, Plus that we know now it's not really the case in terms of how many proteins are generated, because we can tell at that time they didn't know the RNA splicing. Now we know that this sequence, if we just take it and we like consider translating that into a protein, it will give us a particular peptide. So this particular DNA would translate into a particular peptide. Now, the modifications that come at the level of the RNA, which is the RNA splicing, could generate more than one version of that protein. And then, once we have that messenger RNA, the protein sequence cannot be changed. So we'll have that particular protein sequence. The modification that we could do is post-translational on that protein. So if we take this sequence here, imagine this sequence comes from there. So that's a DNA, and we are imagining that this is translated to a messenger RNA, and it's translated to a protein. Okay, so each tree is going to produce an amino acid. We know that we have 64 of the codes, and we have 21 amino acids. Definitely, it's going to start with ATG. We are just snapshotting here a small part of it, which would be that sequence is translated to serine, valine, proline, and tyrosine. Okay. Now, if a mutation takes place and this mutation is silent, nothing happens. So there will be a change. You can see that change happened here in val here. The TA has been changed to TT, but the change in the TT, it still keeps giving you a val, because there are 4 or 6 codes that could code to the same amino acid. So that's a silent mutation, nothing happened. Now we have a missense mutation, we start changing the amino acid sequence. And if we change the amino acid sequence, missense mutation would tell that there's only an amino acid that changed. Now, that amino acid should be translated into a stop codon. And that's when you say a nonsense mutation. And these mutations could cause disease. I gave you examples like Duchenne muscular dystrophy, like sickle cell anemia. So it's only that one base pair that could lead to that disease to happen. The worst part, I would say, is differentiating mutations, because that's a totally different group of integration. Because then you can see serine, valine, proline, tyrosine, and then serine, valine, leucine, and then downstream is going to be everything is going to be different compared to the parental one. Okay. So I hope by today's lecture and the few lectures we have a very good grasp on the concepts all the way from DNA generation to proteins and the levels in between, and RNA splicing and the mutations. And that in the next part of the lectures, I will start doing the opposite sense. I will start discussing, okay, heart disease, let's say. Heart disease is characterized by 1, 2, 3, 4. And then I will tell you what are the genes that are affected. So we'll come back other way around. So the first part of the classes was really molecular. And the second part will be like discovery of how people found out this particular gene causing heart disease or diabetes or cancer or something. Okay, so if a cell is ready to be like it's very prepared to divide in terms of DNA quantity, RNA quantity, proteins, all the nutrients, and then some sort of damage happen. In this particular one, I'm just giving you an example of radiation. So the cell could get that radiation in the upper part in A. You can see the cell had been acquired radiation, but it had the repair mechanisms ready. So it absorbed that radiation and then corrected the error that happened. And therefore, all the cells that are generated downstream of this, they will be normal or look like normal. Okay? The second situation is that the cell has been hit with the radiation, but now it acquired a mutation in this protein. And therefore, every single daughter cell that comes out from that is going to acquire that mutation. Now, if you take this situation and you projected that this cell is a germline, this means it's not only propagating in your body during the time of the division, it will be propagating to the offspring. So that's the inheritance of the genetics. So if the gamete, the egg, or the sperm carries that mutation, it will be carried from mom, dad to offsprings and to the next generations. Now, the third scenario is when the cell is hit with a radiation or damaging agent, but then it simply carries them. It's not evident that the cell is abnormal. But downstream, when more divisions happen, some of these cells will show the phenotype of that division. So the cell will look normal, but it's a carrier. So all these situations we see, and that's why we do genetic tests sometimes. Like when a woman is pregnant, they will say, oh, we want to do this type of genetic test and this type of genetic test, right? So that we could predict if the offspring will have this type of disease. Because we could predict for those, particularly in this situation or that situation. Because if you have it, you have it. And if it's hidden, like as a recessive gene, we could predict that it might cause the disease. Okay, so DNA damage could take place at different levels, and it can be caused by many factors. So I listed some of them for you here, and I kind of categorized them into the two groups. I want you to read the first two lines. DNA damage happens at very high rate per second. This sentence should shock you. Like every second you have DNA damage in your body. So you can imagine how much energy you are spending on the DNA repair. Because you have that. So that would start by a lot of changes. And it's like almost 10,000 to 1 million DNA lesions per day. That's a huge number that your body is taking care of. So what are the sources of that damage? X-ray, toxins, viruses, bacteria, these are exogenous. These are exogenous. Why? This means you are walking here today and you just have to encounter a virus. This is something outside your body. The endogenous sources, these are processes that we need to perform for function. Yet, they would produce byproduct. And that byproduct is going to cause your problem. So this day is going interferences here. Probably that would go to that side. Okay, so the DNA damage, it can take place at the nitrogenous bases or the sugar groups. It can be a single item cut through the DNA. It could be a reaction between two nitrogenous bases. It could be a change from one nitrogenous base. So in the next few slides, I will describe this extensively. Probably there is a little repetition, but it's important to have that very well discussed. So the example I'm giving you here is this enzyme, uracil DNA glycosyl. Well, when this enzyme performs a function by removing an amine group. So this is cytosine, and that's uracil. They are very, very similar. If you look, the difference is only that this has an amine group here, NH2, and this guy here has the oxygen. So this enzyme performs that function in the presence of water. It will deaminate the cytosine. And since water is there, water is, as we said, water is the major component of the cells, and all the reactions has to take place within water. But sometimes these go in a direction that we don't want. Like if we turn cytosine into a uracil, that's already a damage in the DNA. And that's a damage on the nitrogenous base of that DNA. So probably we can correct it simply by taking that out and replacing it back with a cytosine. Okay, so this is a type of damage that happens simply because we have that enzyme and we have water available. So it's an endogenous process. It's not because we have toxin or bacteria or anything. Okay. The second part, it is the free radical. The free radicals, these are products that generate when we are trying to generate an ATP in the mitochondria. Most of them, they have the characteristic that they are able to react very quickly. They have a very short half-life, but they are very reactive. Like I can tell you, I worked with one of these free radicals. It's called peroxynitride, not listed here. The half-life is 4 seconds. 4 seconds. Like when we are generating it in the lab and we want to produce the damage to the cardiac cells, because we were testing how the cardiac cells would react toward an oxidative stress. We had to be ready within the 4 seconds to get that into the cell. And get into the level where this fluctuation of the function is crazy. Like, can you imagine the 4 seconds? If I do it in 1 second, maybe 80% of the cells die because of that peroxynitride. If I do it a little bit slower, maybe 60% die. So they accumulate a lot of changes in the body. If that's at the level of one cell, you can imagine at the level of the tissue or organ out. So the free radicals are formed by water again. And you will see by the end of this lecture that water is there because as we are producing that ATP, which is the energy molecule, you go by a series of reactions. But at the end of the day, you want to stop that reaction. You don't want to have it all the time running. That's where water comes in and blocks the reaction. You will see it in one of the videos downstream. So once that free radicals are around DNA and DNA is negatively charged with the phosphate, like if you look at the DNA structure, you have sugar, right? You have the phosphates, and you have the nitrogenous bases, and all those are like with many-sided chain. So it would be perfect for these reactive oxygen species like ROS to attack certain sites of that DNA and very quickly react to a part of it. So if we see here, the X-ray or the gamma ray will generate extra electrons, and those extra electrons will be donated to water And water has to kinda deal with it. And when the water is dealing with it, it could produce those superoxide or hydroxyl radical. Like this is the hydroxyl radical. Okay, and this is one of the most reactive molecules. Then this molecule is, you know what I call the nowadays the reactive oxygen species? It's the media. It's exactly like the media these days. It attracts you without really wanting to do that. It just sticks to you wherever you go. It's the same thing here. Oh, I have like advertisement. Oh, come to us, eat, and we'll give you another meal. But they tricked you because you didn't want to eat from the first place. Yes, you got 2 meals, but you didn't want the first one from the first place. And that's exactly the same. They have an extra electron, but then they stick to the DNA and perform certain reactions at those residues. Okay, and then recall that these molecules are made in a topology that is very, very specific. So the DNA molecule, it's in this helical shape. Of course, the A with the T with the 2 hydrogen bond, and then the phosphodiester bond on that side. So it's a very well organized molecule. Now you come and add some molecules to that structure, some electrons to that structure. Once you introduce those molecules to the structure, most probably it will distort that DNA. So the structure itself is not going to be the same. And this will make a possibility of breaking the DNA. So you could cause single-stranded break or double-stranded break. So that's how most of the reactive oxygen species affect your body. They would target the DNA. And now we are just speaking a piece of DNA that we are just for a scheme. But can you imagine if this DNA is insulin in your pancreatic cells? You break insulin in your pancreatic cells, beta cells, then you're not producing insulin, then you can't regulate your blood pressure, sugar. So these changes could lead automatically to a change in the transcriptional profile. Or if we go back one step and we think about these breaks happen in the molecule in the DNA that regulate the DNA replication. Proofreads the DNA replication. Therefore, you are not producing the proteins that are supposed to proofread the DNA. So you can imagine this would be very easy to be happening. Because you're not proofreading any changes. It's going to cause the mutation to be permanent, and therefore it will be propagating to the next generations. Okay. So the oxidative stress is produced, as I said, from intercellular sources and extracellular sources. And the 3 major molecules that I want you to remember is the superoxide, an oxygen that has an extra molecule there, the hydrogen peroxide. It looks like water. Okay, looks like water, but it has 1 more hydrogen with, of course, 1.2 electrons, and then the hydroxyl radicals. And these are very well known in the field of medicine for their only of the oxidative stress and therefore cell damage and downstream the diseases. It's written there, excessive ROS production could lead to the cell apoptosis, which is a type of cell death, or autophagy, which means the cell kills itself. Lipid peroxidation and DNA damage. And for today, we are discussing the DNA damage. Okay, but if the cell knows that at certain point the water is going to cause us to production of these reactive oxygen species, then most probably we are equipped with enzymes to take care of it, right? So what are those enzymes? This is one example. Now get ready for remembering your biochemistry. In this slide, you can see we have generated a superoxide molecule. Okay, that superoxide molecule, our cells are very well equipped to take care of it. And the way, remember, we discussed why we have copper and zinc. Because copper and zinc are very important cofactor for an enzyme called superoxide dismutase. So this is a superoxide ion in red. And this is superoxide dismutase, which is here. Superoxide dismutase. Now, this is a very reactive molecule. So we take it in steps. This guy, the superoxide, if I go back, and I give them ranks, but this is not 100% accurate. The most active is the superoxide, then the hydroxyl radical, and then the peroxide. So the cell reacts so quickly to the damage by, oh, we have a superoxide that has been generated. The easiest and the safest way is to call the police. Superoxide dismutase. It will transfer that into H2O2, which is the hydrogen peroxide. And then you have many other enzymes that come to the show in order to bring this back into water. Okay? Am I going so quick? Yes.
Does the peroxisome
Which one?
use the peroxisomes on the H2O, the H2O?
The peroxisome?
Yeah.
It happens in peroxisome, but the enzymes that are doing that is the peroxisome.
Okay.
It happens mostly in there, but it's not necessarily to happen only. They have more of it in that. Now, so how do we deal with the reactive oxygen species? You guys need the break, I can tell. Okay, I'll finish this slide and leave you break. So we can deal with that in two ways. Either we have endogenous enzymes that whenever there's a superoxide hydroxyl radical peroxide is generated, then there's an enzyme that can change that superoxide into something else. As you see, the superoxide dismutase change this molecule into hydrogen peroxide. And then other set of enzymes like catalase and pyridoxine then generate the water back. So this is an enzymatic related assay where we get rid of these reactive species. And we come back to the next part where you guys all take vitamins, and that's why we need to take care of vitamins in our body. Vitamins naturally are antioxidants because they have very complex structure and they can acquire electrons. And these guys, they have more electrons than normal molecules, right? So the vitamins will attract those reactive oxygen species, and therefore they will be deactivated. So by binding a superoxide to vitamin B or C or whatever, then that will deactivate that superoxide, and now you don't have to go through the reaction that we are showing here. Okay, so that's where vitamins come into the show. Of course, there are molecules that are generated, like glutathione. It's a byproduct of an enzyme. So our body produces glutathione, right? And that glutathione then plays a role in the transfer of the electrons from the H2O2 into another molecule generating water, and therefore deactivating the function of H2O2. So either we can deal with it by vitamins and like other products like uric acid, and that's an acidity related process, or we can deal with it primarily with enzymes or a byproduct of the enzyme, which is the GSH. Okay? I'll give you a break. When you wake up a little bit more, we come back for a full discussion on this slide before going. Okay? For those who are online, can you hear me? You guys have any questions? Any slides where are those scenarios where methylation also turns on the gene? Margaret, most of the time the methylation is known to shut down genes, but this is not always the case. There are examples, yes, where methylation turns on certain genes. Or in other words, I can tell that there are some methylation that do not block the expression. Okay, you guys have any questions for the slides we discussed so far? You can unmute yourself. I'm sorry because the setup in this room is not great. I will pause and then I will come back. Okay. You didn't need the breaks because I have the impression that I'm losing you. But my impressions were lost. Okay, so do you want me to repeat this slide again or are you okay? Repeat. Okay. Very quick, so let's go back there. My computer froze, sorry. Okay, so we discussed that certain types of damage starts when we have reactive oxygen species. There are different sources to how these reactive oxygen species are generated. And now our body has to deal with this. It has to correct, if we consider it an error, which is not really an error, but if we consider an error, then we have to correct it. So this is one of the scenarios how our cells deal with reactive oxygen species. Either they have enzymes, and they are listed here like superoxide dismutase. This is the most famous that will convert a superoxide molecule into hydroxide, of course, with other cofactors. And now it's easier to deal with hydrogen peroxide. We have other enzymes like catalase and pyridoxine, those who could take care of the H2O2 generate water out of the bath. The other route is that we have molecules like vitamin, the uric acid, or some products that these enzymes produce, like chloroform, which could attract these reactive oxygen species and neutralize them. And that's how we could, like our cells continue surviving. And you can imagine that this reaction happens almost every other second in our body. Because reactive oxygen species are there, we always need to generate energy, and that's a process that generates thrust. Okay. So that could cause DNA damage, and this is summarized for you here in 5 different categories. The first and the easiest way, easiest form of the DNA damage is the mismatching during the replication. And then these reactive oxygen species, besides other causes of DNA damage, could lead to a single strand break or second strand break, like both double strand break. Or it could lead to the reactions between the different strands in a different way, like as I said, an heterogeneous base as interstrand cross links. It simply reacts to the other strand in a not in a regular manner. And then it would generate these changes in the conformation of the DNA, like the cross links that we're talking about here. I will go each one of these in a slide so that we can understand the difference between them. So as I said, the easiest one is the basic match, because during the replication, you see the DNA polymerase and all the replication fork is generated. Now they are like copying the DNA as moving. If any mistake happens, it's so easy to correct because all the machinery is there. It's similar when you have construction on the road. If all the people in the crew are there, if there's any problem, they can fix it today. 1 week from now, if we have a problem on that corner, we will have to bring all the crew again. So that's why these base mismatches are the easiest to take care of, because the machinery is already there. So this is the list. So the types of the DNA damage dictates what type of repair we have to do. Okay, the more severe, the more machinery we need to have, and the more complicated. And if you look at the outcome, probably we could really get that DNA repaired, and sometimes it is repaired, but it's not in the part. Okay, so here is a little bit more complicated how we do the DNA repair. You can see that now we start looking about many other molecules down here. And I'm just giving you this slide to show you that, yes, molecular mechanism of the disease, it gives you a good review of those things. But if I will go deeper, I would need probably 3 lectures to teach you this slide and start to tell you what's the difference between ligase 1 and ligase 3, ligase 4, and what type of polymerases are recruited in each one of these mismatches. But we are not really specialized in DNA damage here. So I want you to understand the concept. And I added more information at PDF for those who want to go deeper. Okay, so in the single strand break, this means we have already, if we look on the first up here, we have the blue and the red strand. The red strand has broke. Okay, but this is, let's look for the positive side of it. We still have the blue one there. So it's very easy to understand what is to be corrected because we have a template, right? So we have the blue one, we can still read it, and we can simply get rid of those damaged DNA on the red strand, read on the blue, and just complement. So that's an easy fix. Now, if we generate lesions like this, most probably this will cause DNA to break. And therefore, you can look down and you start to see the number of the proteins involved are much more. Because now you have to have more machinery added. I'm sure you will ask me, are we supposed to know the name of? No, you are not. Okay. Then look at the base mismatch. And now you can tell visually that this is very easy to fix. Now, if we deal with that one, it's very easy to deal with that one, because all what we have to do is install. We're going in the propagation of the elongation of the replication step. Oh, we figured out there's a mistake, stop, go back, remove it, and then replace it. And all these are simply changes that we are taking one base pair, two base pairs maximum. The problems are here when we have the double-stranded break. And just let me be correct, sometimes this is much bigger. The stretch of base pairs. While this guy here is definitely one, and here could be only one or two or three. Now, with the double-stranded break, the issue is we have now one DNA molecule. And if we're looking here, you're seeing that now it's two DNA molecules. It's broken. It's two different molecules we have to put back together. So the luxury we had in the beginning, where we had a template to simply remove those and refill, is not there anymore. And therefore, now we have to come and look here. The extremity, so we have to take all that piece of DNA and replace it. And in order to do that, we need to do the homologous recombination. Are you guys aware what is homologous recombination? Did you study that before? Anybody can tell me anything about homologous recombination? No? Okay, I thought you guys knew it, so probably my slides are gonna be a little bit more difficult. Yes?
Is it where, like a similar chromosome? The, genetic, material is transferred to, chromosome like a cell?
Yes. That's where homologous recombination takes place. This is one of the natural situation. So let me simplify before I go to the next slide. So homologous recombination, let's take this situation as an example. Okay, we have this piece of DNA that we are sure that it's a double-stranded and it's templated. So we have the forward strand and the reverse strand. We have a problem here. And then on the other side, we have an intact DNA. So at least we can tell we have two sides where we can use as a guide. So I would tell that if I want to fix this DNA, okay, from this area, I have one guide, and from this area, I have one guide. So I have two guides. Okay. Now, in order to do the homologous recombination, I would have to flip-flop DNA material from one strand to another. It will come in the next slide. But I will I will go slow because your answer is that it's not very good. But I will go slow on that one. So let's start with the first, which is the base mismatch. So here's the DNA polymerase. That's the 5' to 3' end, and it's being synthesized from the 5' to 3' end. It added G, T, T, A, G, C, C till the AT. Everything is perfect till here. But now this is the DNA polymerase and the machinery of the replication. Oops, there's the first mistake is going to be done where the T is added at this part, and then probably the next one is they're going to add a T2. So there's a T added in front instead of the G. Now it stops. It removed whatever I have added and then added a T and continued. Okay, so that's the simplest thing. It recognized that, oops, here I was putting something instead of the T. It could be C or G or A. But the template is A, so the template is dictating that we have to have a T. It stopped, it cuts, remove, added the right, and continue. So that's the mismatch correction. That's the proofreading. And this is called 3' exonuclease action of DNA polymerase. What, like, many times enzymes are dictated by what they do. So if you read the name of the enzyme again, look, it says 3' exonuclease. Exo means cutting the last part, external part. So it's a 3', so it's at the 3' end of that DNA. Exonuclease is cutting that part out, and it's the DNA polymerase. Okay. Now, in bacteria and in eukaryotes, they have different names. I just highlighted that for you. It's polymerase 3 in bacteria, it's delta and epsilon in the eukaryotes. Now, for future, you might hear a lot about high fidelity proteins or high fidelity polymerases. Meaning, this, each 1,000 base pairs, like 1,000 of those, it makes one mistake. Okay? The high fidelity means I would avoid having this mistake every 1,000. Okay, and now we have in labs polymerases that are generated, like by genetic engineering methods, that are one mistake in 10,000 base pairs. So if we have a gene that we want to produce, instead of one mistake in 10,000, we go to 10,000 with one mistake. Okay, so the high fidelity means that the DNA polymerase has a very low error rate. Okay, and this is how we do the PCR, the polymerase chain reaction in the lab. We use these polymerases to generate DNA in the lab with low errors. Okay, so this is happening during replication. Okay, so keep that in mind, that as we are replicating, we stop, fix, and then continue. So this is as the fork of replication is taking place. The next step is proofreading after replication. So this is a mismatch repair. So the DNA has been replicated, the fork has moved, but oops, we found out there is a mismatch. And therefore we need to correct that. So you can tell here, the G with the T compared to the G with the C. So this is only one nucleotide. Excuse me. So this is still one nucleotide to fit. But what happened is that when we have the G with the T, it already changed the conformation of the DNA. So the topology of the DNA chain. Topology change means the stability change. And now, in order to fix that, we will have to add more effort. Because the first thing we have to know is, oh, we have two strands here, okay? And exactly as I told you in the beginning, remember the methyl groups that are added to the DNA? So the DNA is methylated just before getting into the replication process, right? Now, if we have a mismatch, and on this strand, it's methylated, and this is the newly generated strand, which is the daughter strand, automatically I know that this is the wrong base pair, not that, right? Because you want to know which one to correct. So this is the parental. The machinery will look, oh, is it methylated? Oh, yes, it's methylated. So that's the parental strand. So therefore, the T has to be removed and replaced with the C. If we don't have that methylation guide, we could have removed the G and added a C on the other, and added a T. an A, sorry. And the T will stay there and it will cause a permanent mutation. You got that point? Okay. And of course, there's different machineries between eukaryotes and prokaryotes for that. So then we come to the third type of repair, which the damage now is not because of the DNA sequence, it's the damage that took place in the structure of the nitrogenous base. So it's still a C in the right place. So it's a G and a C. But probably that C has some chemical modification like alkylation, hydroxylation, and so it's not a functional C. So you want to remove it. But then you can tell it's going to be more or less the same situation. First, we have to detect it, that oh, the C is not really in the nice chemical structure. And then you can treat it as a one nitrogenous base, like a nucleotide. So we'll have to remove that nucleotide away. So first we do in a step that we have this enzyme, I just wrote it down. Apurinic and pyrinetic, where you have just some changes that causes the nitrogenous base to be chemically changed, and therefore you want to take that out. So you consider it as if it's a whole nucleotide, but the problem now is not in the sequence, it's in the structure. And one of the issues that could come out of that is most of the time when you have like many E and you have a modification on those thymidines, then it's represented for you. It makes such a distortion in the DNA. But now when you fix it, the DNA structure will go back. You can imagine that this T and that T could make more hydrogen bonds together, or the hydroxylation had more charge on one of the thymidine compared to the other, and therefore the DNA structure on one side had been changed. So base excision repair is nothing more than just taking that nucleotide out, but the cause of that is the change in the chemical structure of the nitrogenous base. And you can see in the structure written in the diagram, these enzymes will recognize that nitrogenous base that had an issue, and then it will be excised. So now you have one base pair to be replaced, similar to what we discussed before. And now the same enzymes that perform the addition of that nucleotide, they will do the same. Okay, the fourth one is the nucleotide excision repair. And the nucleotide excision repair is not necessarily happen on only one nucleotide. It can happen on the stretch of many nucleotides. And this diagram shows you a damage that normally happens in the skin cells with a lot of UV light. When the UV light hits the cells, then the thymidines, they will make dimers. And these dimers will definitely change the structure of the DNA. And if it's not fixed, it will cause single strand or double strand break. So therefore, when this happens, it mostly happens where if it says thymine dimer, this means this is a region of the DNA that have many T. Okay. And in order to fix it, our cells excise a piece of that DNA. Excise before and after, and then replace it. Now you can see that we still have that template strand that we can use. So you excise all these T. Of course, if it's a T on the upper side, then it's an A on the lower side. So then you can simply replace it with T. Okay, there's one thing that I'm
forgetting to say in each of those. Here we don't, like here, when you take a nucleotide and you try to put it back, you need to do the ligation, you have to do it. So all the time you have to use the ligases. It's written in the slides. And you can see here, the same thing. You need the polymerase to come here and add the nucleotide and then the ligase to glue it. Then the same here. You added all these nucleotides back, but then you need the ligase to glue it back to the mold. There's a small video. Maybe it's like a short one. I don't know how many minutes is that.
The DNA in just one of your cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times, nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of damage. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA backbone. Double strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two complete double-stranded segments.
So that's the homologous recombination. I will repeat it one more time. You see what happened? The lower, so the lower DNA in pink was intact, right? So you could see the damage that happened. It unwinded and it took that part as a template. Just watch it again one more time and I will explain it again.
Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes
Okay, so I want to stop here. So you see, how did we know which part of that DNA we can use? Because this part of that DNA in the blue is exactly the same as that one in the pink. The intact part of the blue is the same as the intact in the pink, right? On this side or on that side. So now we can make bridges here that this area and that area are doesn't need to be repaired. The repair has to happen within this area here. Okay, then how can this pink serve as a template? It has to be cut. And that part of the DNA will use the lower part as a template, and the upper part is going to be the template for that upper part. And you can see the exchange happening just in the next few seconds.
relates the damaged.
So you see, it has a cut at that blue one. And the next step is that it's going to take
undamaged strand.
the upper band, the upper level of that DNA as a template. Now you have kinda make a replication of this small part of that DNA, just to make a replication of it. And then you can see, because there's no information that will come from the blue. All the information has to come from the pink, because it's the intact one, right? Now you have unwinded that thing, and then you are reading it and you are duplicating it. But in order to do that, you have to first cut it and unwind it and then make it continuous with that blue signal, okay? So then
Strands, get them to exchange
you see, you just duplicated that pink, ligated those two parts, one on the upper blue and the second one in the pink.
sequences of nucleotides, and finally fill in the missing gaps to end up with two.
And what do we have now? We have an exchange. Because the continuity is that this lower part is the template for generating a new blue that was not there, while that upper part is already bound and becomes a part of the blue, and it's the template for the one that was damaged on the other mode.
Two complete double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available.
You see, it's just raised, where's the template? Did you have a template to read from? Didn't have a template in this situation. And that's why most of the time the double-stranded break could lead to cell death right away. Because there's no way to repair that DNA. And even if it's repaired, there's no template, so you're not sure.
Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve, but most of the time, we want DNA to stay the same. Defects in DNA repair are associated with premature aging and many kinds of cancer. So if you're looking for a fountain of youth, it's already operating in your cells, billions and
Okay. So then the next type of the DNA repair is the single-strand break. Now with this video, I think it explains much better than I do. The only thing I want to add is that every type of these repair requires different protein. And as researchers, we always try to understand what the protein does. And for example, this protein PARP here is specialized and it's upregulated, so it's produced so much whenever there is a lot of single-stranded break. Because it is the first defense mechanism to bind to that single-stranded break in order to keep that DNA intact. And therefore, it will direct that machinery of the polymerase, ligase, and the repair mechanism to take place at that site. Then the two homologous recombination, like they are already described in the video, but as it said, the double-stranded break are the most severe because simply the proteins will come and bind to the ends of that DNA, and then they will recruit other components and then try to fix whatever they. And then the generated DNA most probably will have mutations or missed DNA sequences. And that's why most of the time it causes cell death. Now these operate differently. Like you can see the non-homologous recombination and joining, it's throughout the cell, the cell cycle. This means it's always running to check if there's double-stranded break effects. While some others, they are like the homologous recombination itself, it's mostly assessing during the synthesis phase or the G2 and during the mitosis meiosis. That's mostly where it's active. Okay, so I hope this gave you a good overview of overall the DNA damage and the repair mechanisms that take place. And obviously now you can relate how these DNA damages could lead to changes in the gene expression and therefore the function of cells and tissues and cause diseases. Now there's a list here. There is one error I could spot there. It lists for you the DNA damage. It lists for you what are the types of the repair and the diseases. And I don't really want you to memorize what type of disease come from what type of DNA damage. It's more just an example of an application, how we describe one molecular changes that could cause a disease. What I want to show you is this video here, which is an introduction for the metabolic disease, in fact. Okay, where is it? Okay. Okay, so all what we described about DNA damage, we're gonna just narrow it down now to mitochondrial disease. Okay, and it will be the introduction for the next couple of slides, and probably I will stop after that.
Mitochondrial diseases cover a wide range of conditions caused by genetic mutations. In the UK alone, they affect thousands. Mitochondria are the powerhouses of the cell. They use a process called oxidative phosphorylation to generate energy. But a mitochondrion is a complex piece of machinery, and a lot can go wrong. It takes around 1,500 proteins for mitochondria to function, and problems with many of these can lead to disease. The cell's nuclear DNA creates all but 13 of these proteins. The rest are coded for by the DNA in the mitochondria themselves. And mutations in either can cause problems. A cell has just one nucleus and many mitochondria. All the cell's mitochondria contain their own DNA. If a mutation affects mitochondrial DNA, some might be healthy, others defective. And different cells can have different numbers of defective mitochondria inside them. That means that some cells may be less affected than others. The diseases caused by damaged mitochondria can also be varied. They can affect any organ or tissue, and they can appear at different times throughout life. For example, Leigh syndrome is the most common type to develop in childhood. It can affect patients in a number of ways, including loss of skills, epilepsy, and problems with muscle function. Mutations in over 75 genes in both nuclear and mitochondrial DNA can lead to this syndrome. So what can be done to tackle mitochondrial diseases, like Leigh syndrome? Treating patients is difficult, but in some cases, the disease can be stopped before it's passed on. Mitochondria are inherited from the mother. If the mutation is in the mitochondrial DNA, its inheritance can be blocked. One approach is to combine the nuclear DNA from an affected egg cell with healthy mitochondria from another woman. The embryo would then have nuclear DNA from the original mother and father, and mitochondrial DNA from the egg cell donor. This has led some to call such potential infants three-parent babies. This technique is still very new, but it has recently been approved in the UK. It may be a controversial approach, but techniques like these could finally help combat these devastating diseases.
So this is one of the applications, and did you understand that last piece of information that such diseases, what they do is it's just it's not new, it's really approved. It's much more difficult than individual fertilization. So you are taking the genetic material of that nuclei in one mother, and you are doing it in another mother's cell, and then that gamete is gonna be the producing embryo. It's a lot of work. I would say it's scientifically proven that it works, but we kind of overestimate how much we know. That in vitro fertilization itself is very okay. So can you imagine that one? Extracting just the nucleus out into another cell? That's too much. So anyways, This video was amazing to show you how you could come trying to solve a disease, but it could fall into controversial and ethical problems. And obviously, we are speaking about cells, nuclei, mitochondria, organelles, as if they are separated. But the reality is not that. I mean, in the lab, if you will try to isolate the nucleus out of the rest of the cell, it's gonna be like super tedious process, like never 100% pure. So you will always carry mitochondrial proteins or genes with you. You will carry endoplasmic, cytoplasmic related. So it's important for you as future medical provider and professional in this field to always evaluate the ethical and the practical aspects of what you're doing. And this is a very important component to this lecture, in fact, and to other lectures. That yes, we think we know how DNA polymerase work, but after 3-4 years, we get a research paper that negates what we have thought for 1 year is correct. And if we have created a vaccine or some sort of drug to treat, maybe tens of thousands or millions of people have already been affected by the time that we discover that it's not the right treatment. So you always keep that into perspective and think a lot about the practice and think a lot about who is taking advantage of that practice. Because if it's a company, a medical related like drug synthesis company that is generating those and trying to tell public like, oh, it's gonna be the best to go for that round, it's gonna make less burden on the society and it's gonna make less economical a number of billions of dollars you spent to treat those patients. You are the people who have to evaluate that in the future and say, no, it's not the right therapy. It's not gonna work. Or if it affects 5% of the population in a negative manner, these 5% are still human. You can't simply get profit from treating 95% and killing the other 5%. So you have to be really aware of these small molecular aspects that at a certain point in your work is gonna turn out into ethical problems and society related problems. And you are the people who know. People in the street wouldn't know that. So you will be the educators and you will be the person who are in charge. So take very good care of what I'm saying, because we see in this society and in the medical field that there's a lot of mistakes that have been happening. And companies are really using the advantage and the power they have in order to push using the drugs and get those approved. But then at certain point, there's a lot that's affected. Okay, so with this, I will introduce 2 more slides and I'll set you free for your next week of exams. So after discussing DNA replication, DNA damage, protein synthesis, RNA synthesis, all that. Now we're gonna move to the cellular aspects. How will the cell start reacting to the loss of the DNA? How the cell would react to the loss of a certain protein and mutation of a certain protein. So in the next few classes, as I said, we will start describing diseases in the backward steps. And probably the only last part that we didn't really touch on is the metabolic diseases and how the mitochondria generate those ATP. And how would that affect the reactive oxygen species that are being generated. And when the mitochondria would fall into the situation where it has to divide or not divide, or it's damaged or stuff like that. So I'm not gonna go for all the whole slides. But in the first lecture after your reading week, we will finish this and start with diseases and the other aspects here.
Value from here on out, this will be,
The last slide I will be describing, it's gonna be,
oh, these two.
like, again, probably we can discuss a few minutes about the midterm exam before going today, because for example, this slide, I'm not gonna ask you about how you relate that telomere changes into which type of repair and which type of disease that happen. I could, but pretty sure 1 week after you finish your exam, you will not remember.
So this life that we're not something possible beyond religion.
I said, today, we will cover, yes, they will not be. The last slide I described, it will be after that. Even if it's in the lecture, it's not included in the exam, because I will have to attempt to examine you if I don't do this. Okay, so whatever happened at the level of this molecule, it could lead to a cell injury. And that could be categorized into 5-6 different themes. Well, eventually, the cells, they will go to adaptation. And that adaptation, now we know it is, oh, there's a mismatch. And the protein machinery, the replication machinery, or the DNA repair mechanism are trying to fix it. That's the adaptation process. That fails, the cell has to die. That's the best scenario. Okay, so whatever we have described in terms of the stress on the cell that shifts the cell from homeostatic balance into adaptation, the cell is hanging there, waiting. It can stop all the processes and hang there to fix it. It doesn't work, the best scenario is to die. And now we can tell, like these are really vital signs of the cell. Like if the cell doesn't have ATP, the generation of the ATP, adenosine triphosphate, this is the currency. It's like living in Ottawa with no dollars. It's impossible. So the ATP, if it's not there, you can directly tell that there's an issue in the mitochondria. Because ATP generation is mainly mitochondria. Okay. Now we can go back and start tracing what's the issue. Is it a mutation at the genes that are needed for the ATP generation? Is it a protein issue? Is it a membrane issue? Which brings us to the next. Membrane damage. We described how these lipid bilayers make these cellular barriers in order to define what is the cell and what's outside the cell. So if there's a damage in the membrane, internally or externally, if the mitochondria has a damage, the mitochondria is not going to function. And therefore you will not have ATP. Now, if there's a damage in the cellular membrane, now the cell is open, it's going to burst out all the component. So there's no barrier. So that's a sign that the cell has to die. We discussed extensively protein misfolding and DNA damage. Like protein misfolding, remember we have to go for the 3-4 structure, primary, secondary, tertiary, and quaternary structure. If the proteins fail to go to that step, we are producing a lot of protein, but they are not functional. Therefore, if I keep producing those proteins, I'm losing all that energy or producing proteins that do not really help. And at certain point, this will trigger the apoptotic process. I'm talking a lot, there's not information, I can tell in your eyes, where is the text? He's talking, where is the text? Don't worry, it's gonna be there when we discuss the types of cell death. It will be listed one by one. But sometimes I like to talk to you and look in your face, because it's like too much just putting slides for you to read. Okay. Mitochondrial damage, it's very spoken to that. Mitochondrial damage, there's no ATP. Now, we didn't really cover much that one of these components, like calcium, increase in the cell. Calcium increases so much, it's positively charged. The positive charge is added on the cell. And calcium is very important for the contraction of the cell. We needed that. But if we increase it so much, it could cause that the mitochondria swells. It will swell to a level that it can burst. And then you can fall back into mitochondrial damage and then loss of ATP. And therefore the cell death. So you can see they are very well connected together, but they are characterized in a sense that one event of those could trigger the cell death. Like you generated the reactive oxygen species because you have X-ray or gamma ray exposure. Then reactive oxygen species are going to affect the DNA, damaging it, changing the transcription of certain genes, therefore the proteins are changed. If those proteins are needed for the mitochondrial function, the mitochondria is damaged, and therefore the cell dies. But each one of them can reroute back all the way to the molecular aspects that we described. And of course, this explains to us that how we could trace back those types of diseases. And it's important to know what's the cause. Because there's no way we can fix my membrane damage. We can't do that. Membrane damage, this means it's done, the cell is dying. But if we have a mutation, nowadays we have CRISPR, we can fix it. We have the CRISPR technology, we can go and change that mutation. So some of those we could still fix, others that's a one-way ticket. There's no way to fix. Okay, and I will just introduce this last slide, and I will let you read about it, because this is one slide or two, wait, two slides, sorry. So the notion of the electron carrier. Have you guys studied metabolism before? No? Yes? Yes. No. Electron carriers, how the electrons play a role in the generation of ATP. Okay, some of you yes, some of you no. But I will go back with you like the first lecture. Remember when we described the molecule from the beginning? And how this molecule plays a role in performing the function. Like the electron carriers, I wrote it down here so you don't get shocked. It's a small compound that function by shuffling electrons. This means I can have someone who is very green, very green in the room, who will take any electron that is available. Okay, and I would have some who is sitting and is accepting that electron whenever it's available. Okay, so this means there is someone who likes to capture that electron, and that's an oxidizing agent. And there's a person who have an electron that could donate easily. So if we look at the equation and we go back to describe it, we have NAD plus, which is always ready to take an electron. So it's an oxidizing agent. Okay, and we have a reducing agent which could donate that electron. Okay, but now whenever the NAD takes that electron, the beauty is that this has to donate it. Yes, it's ready to take it, but it has to donate. So it will donate, it's very like, I can tell you, if this is a politician, this is giving the electron to his family. He is getting positions in the government for his family. So this is NAD plus, is gonna give the electron to NADH. And then NADH is like reduced, it's in a stable state, and it can perform the function. Okay. So these compounds can easily get reduced. So accept electron, and then oxidize, and maybe lose the head. Now, NAD, which is the nicotinamide adenine dinucleotide, is derived from vitamin B. And again, think about your vitamin. So that's a very important source. And NAD can accept electrons from like any other organic molecule. Okay, so that's good. Why do we need to care about that? Because the NAD is coming here, the NAD plus and the NADH are practically the molecules that generate your ATP in the mitochondrial membrane. Okay, so it's important that when these electrons are removed from a compound, that compound becomes oxidized. Sometimes this is really tricky. You have to, I don't like organic chemistry, like people feel like they start to change names. Like this compound is a reducing agent, but it becomes oxidized. Why? Because somebody told that electron out. Okay. And a compound that oxidizes another is called an oxidizing agent. You would see this in text. I'm sorry, it's not me who wrote it. This is chemistry. So this oxidizing agent oxidized that reducing agent by stealing its electron, and then this oxidizing agent now it has an electron, so we can keep going in the process. NAD plus could give that electron to NADH. So now it's playing the role of reducing agent. Well, that's great. So we have the oxidized form and the reduced form of NAD. And of course, we have the NAD too. And these are the two major components of our mitochondria complexes to generate the ATP. We described in the first few lectures how the ATP works, its function. I'm not going to go to that. I will quickly just show you one last video, and then you guys are free. Okay. Probably I went so quick, just the difference between the two structures I just highlighted for you here. Okay. It is.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double membrane organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane.
So recall, inner mitochondrial membrane. So the mitochondria has two membranes, and these complexes are sitting in the inner side of that membrane.
One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix.
And this is one of the exam questions. Where is H plus more, inside mitochondria or outside the inner membrane? So
The membrane also contains a large protein complex called the F1F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4.
So if I stop it here, so you see the protons are entering only through these complexes. So every time the proton comes in, it has to donate the electron. And the cell decides how to use those electrons. And you can see those molecules here are carrying the electrons to the ATP synthase. So that's how these 3-4 complexes help in generating those. And this guy here, ATP synthase, decides how to use them. Okay.
Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain.
Yeah, the electrons, like this means the 4 are using that chain of transfer.
Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers.
So here NADH come to the show. It's ready to donate those electrons anytime. While NAD plus is going to take those electrons. So when NAD plus gave the electrons to NADH, it comes to that complex, it easily gives them.
Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center.
So you see the organization that as you go down, this molecule has the affinity to take the electron, but this has more, this has more, this has more. So the electron doesn't have a chance except going through all of them. The last one is the most attractive.
A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule.
And that's how it transferred from here all the way to ATP synthase. So we used across the complex to get H plus pumped from outside to inside. But the electron has to continue going somewhere. The coenzyme grab that electron, and now it can go all the way to the synthase.
Complex 2 is similar to complex 1 in 2 important ways. First, high energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality, they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
Okay, we'll stop here. And hopefully tomorrow we have this session for the exam, question and answers. If you have any, if you have any question, I will stay here in the rest of the 10:30. I'll see you tomorrow. We have to do that. All exams, not only mine.
You can also, if you want to vote because of your shift, I think that's. That's all.
Yeah. I don't think it's structural. I know the leaders one, and the small one, I know the charge one, but I don't even remember the problem. Maybe other professor, is passing from other. Maybe not this, maybe some students not to do it, but actually might. But they don't, you know, I don't. I'm sure to be. I focus more on the concept. And you can see the explanation, how it's built, and but the real concept, the concept. But yes, sometimes it is done in a private name or something like that. Factually, that.
So we asked, so into Suman arguments,
Oh, yeah.
it's, yeah, it's not totally right here.
Have. Well, the interest rates could be justified. Anything that we discussed.
Okay.
The interest rate, you could imagine was affected by anything you do. You have to decide that.
Okay, so that means.
Yeah. The fact that it was the real one is because they keep find out. Crossing on the wrong thing. So the way to repair it.
Hi.
and-
Yeah.
The
It's over and way faster.
I think it's gonna be like 61. Or mix of and
Right. Very soon.
But I will announce. Oh, you will get an email on Friday. I will be submitting the exam to the center. I will send you an email telling you what the. You might, if you need it. Yeah, you need the camera, just see how.
No,
The quiz is unrated, it's just for you to click, it will tell you go to responders, you click on responders, and then you see that question, so I just want to go with that process.
Yeah. Thank you for that.
It could be, I didn't say, it should be an easy exam, don't worry, guys, just if you were in
No, I
class,
just-
study this class.
Yeah. Stuff.
No, if you're there.
And what those are, is the point.
But
Yeah.
also, if you don't even.
Yeah.
If you are with SAS, you're welcome to do an exam certificate, I reserve it for you. But. You want to, then decide to do it at home like everybody. If that's. Yeah, and give you the extra time, of course. So beautiful. If you want to, yeah. For each student, I went, information and I remember.
So they're not being released.
If they expect to, they're not gonna, don't worry about that. If you don't show up, you don't show up.
All the release.
Yeah.
And then if we're going to touch them, you know, but if I'm with all, no, because. Yeah, but we.
I don't know.
Or,
Which one is this before or after?
this is right after, this is the video, and then that's the slide after.
So we can stop.
Okay, so,
This
so is this included?
Yes.
Okay, Okay.
But are you negotiating on that? For you, I'm not ready. Okay? Let's go there.
Sorry, can I see that too, which slide is that?
Yeah,
Like
I'm
what?
gonna,
Gotcha. Yeah.
could you please screenshot this and email it to me?
What,
Screenshot,
this one?
yes?
Not this one?
No, this one. Okay. Screenshot it and send it to me and say exam,
Okay.
okay? So I don't go to that other one and we got in trouble with that.
Okay. Also, I think. That was your point. Ask about this, but for people that are trying,
They get extra.
do we do it at, like, at our home, or do we do it at the exam?
I, like, for people who want the implementation, I
Yeah.
try to do the maximum. So I reserve for them, the exam center. It's up to them.
I see,
They feel themselves, they do better there, go there.
I see,
You wanna do it at home, you get the extra time.
okay,
So I try to recommend it.
okay, so because at the exams, I'm doing brain, like, I'm not sure about stuff.
No, no. But I wrote it down, though.
Okay,
Um,
it's
it would be proper, but
Bell Proctor. On there, that's why, okay,
we don't need to do that. So.
yeah, okay. Awesome. Thank you.
I just have a question, I about, since that needs to be examined. I know some projects and most of the slides, and then I was, textbook, and most of the time, I'm just wondering for your class, it seems like it's mostly.
Majority slides,
Okay,
if you find yourself struggling because the slide text is like crawl or something, very long.
right,
Go to those PDFs now. They are not very long, they are page or two minutes.
yeah, okay, so.
But majority is the.
Okay, because even the video you showed us, where I can't even explain right now, but where they were replacing the vote with DNA and crisscross it, that whole thing, like, I probably need to brush on that a little bit.
Yeah.
So I guess the PDFs would probably go for that, right? So, okay, all right, so if we go with that.
Yeah, I do two PDFs that all have the same content on the DNA damage, one of them has better images, but the content is the same.
Gotcha, okay, all right, so I'll take a look at that. And the other thing I wanted to ask you was just something that you got me to go with the kind of the treatment options, and you said there are certain, I guess, methods of force might work for 95% of the population, but the other was quarantine and complete that. I'm just kind of wondering what you mean by that, because I feel that there, in every treatment, there's a certain group of people that are for, like even when we saw the COVID vaccines, right? There's a minority that actually got disadvantaged. So I'm just wondering, what do you think the.
I think what my point is, sometimes we do research about substances, and we come to a conclusion that this is 95% on that is gonna change.
Okay.
And if you read this sentence, it means that 5% will not change.
Right, true.
That's the statistics we use.
Yeah,
Sometimes biological effects doesn't really relate to functional. Like if you say this is significant, meaning the numbers in terms of mathematics are different. Like these are human beings. You can't say it's significant.
Yeah.
And go and practice on them like a test.
right.
So what I want to say is that when you get many times the vaccines or drugs, many times they publish that it didn't work on those people. And they were people in the case study, right? So, but then still they ignore it and it gets approved,
Right.
right? So that's my concern. Like when you publish something about a new drug or a new method or something like that, you have to acknowledge that the rest of those people might be affected. And I'm sorry to tell you that I don't see that happening.
Yeah, it's true. It's true.
That happens when there is a withdrawal in disease after 10 years, but how many years were affected?
Yeah,
Because you ignored,
yeah,
like that study that you took 100 people, it worked on 80 and it didn't work on 20%. And then to find a reason to interpret, say, oh, these 20 people, one of them was genetically not the same, one of them was ethnicity different, so you find reasons to say it works.
right.
but it doesn't work on this.
Just ignore this small group of people.
But then when you go to sell the drug, what happens? Your medical doctor is not gonna look at your color or
Yeah.
ethnicity. He's gonna not remember that study that came out 3 years ago about how this,
Right.
right? You're gonna say, okay, he's having a symptom of liver cirrhosis. So what's the drug do you guys prescribe? He's gonna prescribe.
Yeah.
And then it's gonna affect that person in a negative way. So that's exactly like healthcare providers.
True. So then do you think the solution is just don't approve the drug at all, or do you think it's to regulate it?
To regulate it
Right.
and have it more categorized. And that's what, nowadays we have what we call precision medicine.
Yeah.
And precision medicine has lost, well, it's still very far to reach the point. Yes, then aspirin cannot be given to all people. Sure, Tylenol cannot be given to all people. Yeah, Advil cannot be given to all people. So that's what,
There's a lot of like exceptions.
and you guys are the one side.
Okay, okay, I think I get your point a bit better now, because before I interpreted it as, oh, just don't approve the drug, period.
You can delay
Yeah,
to get more information, that's a good thing to do.
yeah,
But you should also look at the benefit. So if it's beneficial in case of the COVID-19 that's affecting like millions of people and dying people, then you really want something as quick as possible to the market. But it doesn't really mean that you have to rush it to a limit that you don't know the side effect.
yeah, no, totally, I get that.
That's how we should work.
Yeah, okay, all right, awesome, well, thanks for explaining that to me. And yeah, I guess I won't see you next week, because the exam's online.
Yes, and then you didn't be right.
Yeah,
Well, good luck with everything.
yeah, thanks, I
I'm
appreciate
sure
it.
if you let the police back.
Yeah, I won't be able to make it to the practice session tomorrow, just because I have another commitment, but.
Yeah,
I'll
but
try to
I
watch
would
the
post
recording.
that, yeah.
Yeah, I'll,
Everybody have a chance.
for sure, yeah, okay, awesome, thanks, take care.
Alexandra? All good? Not really? Why is that? Why is that? Oh, it's still recording? I can trim it later, that's fine. Guys online, you still have any questions? Okay, no response. Well, thank you very much. Have a good day.